Well, hello, and thank you very much to the organizers of the annual UKAFP conference for the invitation to be with you today. This is quite an honor to be speaking with you all, and I certainly wish I could be there with all of us wish that we could be together today. And it's a pleasure to be able to speak to you about the impact of viruses on wastewater as we think about advancing public health and as we reflect back on where we have been in terms of water and viruses. So my name is Kali Kniel. I'm in the Department of Animal and Food Sciences at the University of Delaware. And this is where my office and lab are located here on the left-hand side. We have a lovely farm directly on campus where we produce milk and cheese. And we're able to do a lot of research there in the farm as well. And Delaware is nestled between New York City and Washington, D.C. So I'd love for you to come and visit us. So when we think about wastewater, we think about viruses in wastewater, we're referring to a branch of science now that's called wastewater-based epidemiology. And this is where we are looking for trends associated with disease. And when we think about sampling and surveillance for viruses in all different types of water, we're really thinking about the impact of populations on those different types of water. So for example, in wastewater-based epidemiology, we're thinking about perhaps viruses that are shed by humans within specific communities or specific populations. And we'll reflect back on how we got to where we are today and how useful this complementary approach to understanding the variability of the virus SARS-CoV-2 in our environment is and how complementary that is to clinical testing. If you look at this infographic here on the bottom, you can see that we really have been using this type of epidemiological approach for some time, using it for various analysis, using that in comparison to other types of data. And then a lot of methods development has gone into all of these different studies that we're going to look at today when we think about people's use of collection and identification of the viruses or different chemicals, such as those that are shown here on this slide. On the right-hand side is the new center that we have at the University of Delaware, and we call it SEWER for short, Center for Environmental and Wastewater Epidemiological Research. And it's really a pleasure to be able to share some of our data with you today as well. But let's back up a little bit, shall we? And think about viruses in water and all of that, what that means to us. In terms of food science, we are typically thinking about shellfish and berries. There are various methods that we can use to test to see whether or not these products are safe to consume. We can also look for viruses for surveillance. And many times we ask questions about which viruses should we test for? Should we look for indicators of fecal contamination, for example? Or should we look for surrogates so that we can test those in a processing environment and use them for validation, for example? Are we going to be doing environmental testing in waters like our marine habitats for these shellfish, or are we looking for a way to ensure that sanitation is done properly? This paper here on the right, which just came out fairly recently, is looking at the global question of rotavirus, a very important virus, especially in terms of the health of children around the world, and trying to understand where these viruses may be and how they then interact with people across the globe. And so viruses in water can be a very large topic. This is a great paper that's looking at indicators for waterborne pathogens. This is a historic document that was written by some of the greatest minds in water and viruses that we've known here in the United States. And they looked at a series of outbreaks that occurred and of known and unknown origin here. And they tried to track those and understand what that meant in terms of what was happening in those periods of time. And so we can have a historical approach when we think about waterborne pathogens. I have a couple papers here that are fairly recent that I want to show you the breadth of different research that people are doing when it comes to public health and viruses in water. Here, this is a study that's, again, going back to looking at SARS-CoV-2 as well as variety of other microorganisms and trying to understand how can new technologies like next generation sequencing, how can we better understand what's going on in these complex wastewater systems and really understand what's happening in terms of microbial community analysis. And so very exciting work that's being done here. Also, if we look at water with a One Health lens, if we think about how important that triad is where environmental health impacts human health and impacts animal health, and how we really want to have a lesser disease burden across all three of those circles. This is quite an interesting paper where they're looking at climates and seasonal influences in terms of the ecology that happens in various countries, here in some cases in Africa, and they also extend this to other continents as well, and trying to understand how the climate might impact ecological changes that then impact the presence of viruses in different types of water. So really interesting ways to think about viruses in water. That brings us back to some of the work that we have been involved with in my laboratory, and increasingly in the United States, farmers are trying to understand what types of water they can use to irrigate their crops, not just grain crops, but also fresh fruits and vegetables. And so we have open bodies of water like the one shown here on the right-hand side. That's a beautiful river, and there is a grower who's actually pulling from that to irrigate corn. But what if this individual would like to use tomatoes? Well, how can we best understand what the microbiological quality might be? Well, Dr. Anderson Coughlin here, who's shown here on the right-hand side, she was able to use some of these historically used methods and be able to optimize them for use in surface water as well as reclaimed water, both water types that are often very full of particulates and very high in organic load. And so difficult to use traditional filter techniques where we try to pull out the viruses. So in these studies, what Brianna found was that in looking for the presence of enteric viruses, she found in many cases, especially in the reclaimed water here, if you looked right through the center, these are enteric viruses that were found here, found a larger percentage that reddish, purplish color here. There were more of those in the reclaimed water than there were in the surface water, and not very many human pathogenic viruses in the surface water. If we try to compare that with an indicator virus like pepper-mild mottled virus and try to make sense of whether or not the presence of that as an indicator might correlate some way to the physiochemical aspects of the water or the type of water, we really didn't find great correlations, but we made some analysis, and we have this paper coming out soon that was pretty interesting in terms of that next step of trying to understand how human norovirus gets into these water samples, or something like hepatitis A might be in these water samples. So now if we turn to where we are today around the globe, we have really been dealing with something that has been a changing target. And in many ways, that's where we go back to history and think about what we know from viruses in water to be able to have additional tools in our toolbox. And this has really been a moving target, as I mentioned. On the left-hand side, you can see some information from November, and then on the right-hand side, this is from March 1st, looking at the number of cases around the globe. So what happens when we think about detection in wastewater of SARS-CoV-2, right? We know that this is a respiratory disease. COVID-19 is a respiratory disease. We also know that there's been millions of people who have been infected. And so we have this high percentage of learning a lot about this virus in terms of the way it infects different individuals, and also how much virus that we might have in different bodies of water. And many scientists are working diligently to try to make the best correlations and connections with all of this information. So we know that SARS-CoV-2 infects the respiratory tract mainly through this binding to an ACE2 receptor, while ACE2 is also highly expressed in the intestine. And there have been some studies, especially out of Asia and China in particular, where they've been able to identify individuals, one individual that perhaps the fecal containing virus was infectious to cell culture, but in many of the others, the virus was not infectious. And there also is a chance that the intestinal infection can contribute, at least in some patients, to what's known as that cytokine storm, that release of cytokines that may also be involved with viral pneumonia and some of the other severe symptoms that occur. We've seen a variety of different amounts of RNA that can be detected in feces across the globe. And again, getting back to that first point, with so many millions of people who are infected, there's going to be a wide spread of data that comes from that in terms of clinical evaluation. But let's keep in mind that it is highly unlikely that these virus particles are infectious when we detect them in wastewater. And that is in part due to the fact that SARS-CoV-2 is an enveloped virus. So just a reminder that in food science and as food safety specialists, many of us are used to thinking about how to control very hardy and very difficult to control viral pathogens like norovirus and hepatitis A virus. Both are non-enveloped viruses that are spread by fecal oral transmission. And we know those are environmentally robust microorganisms. But we do not see that at all with SARS-CoV-2. In fact, that envelope actually makes it more susceptible to disinfectants. That envelope that surrounds the coronavirus is important for its ability to be able to bind to the host and to be able to infect that host. And this is an infographic that we designed early on in 2020 to help people understand those differences and thinking about why, if it has an envelope, why is it so much more susceptible than those that are non-enveloped? And so we hope that some of these images here can be helpful to explain that. So let's get back to wastewater-based epidemiology and talk a little bit about the history of WBE. And again, we're looking at the ability to analyze a community through fecal and urine samples. And what's really nice about that is that you can pivot if you need to in terms of mitigation efforts that need to be applied, and you can help various sizes of communities. And looking at these smaller catchment areas has been something that is very exciting to see that science is really digging into in different parts of the world. So when we think about the big picture, again, we're looking at temporal changes in virus concentration and looking at diversity of population that's excreted in this community wastewater. That diversity of population of viruses can also be useful when we think about detection of variants of concern. And now we're looking at wastewater to be able to identify where those may be found. So in combination with monitoring metabolites or biomarkers, scientists are able to make adjustments for different populations that might be there. Also using this technology to be able to identify before a clinical outbreak happens, when you see a spike in the amount of virus that is being shed in a community. And of course, there's a lot of thought that goes into this. Careful design of sampling routines, thinking about the location of where you sample and how frequently those samples are collected. So over time, a variety of substances have been tested for in wastewater and have been used in terms of this epidemiological approach. You can see some of those here on this slide, from alcohol to different types of drugs, and these have been tested for around the world. So this is certainly a technology and a technique that's being implemented in many places and has been for some time. But there's always been this focus on virus detection. And historically, we've used epidemiological approaches to wastewater to try to understand how to reduce the disease burden. And these projects have been funded by a variety of different funding sources, and again, been employed around the world. And the focus on viruses is really because we have so many unknown diseases that we know are associated with viruses, but we just are not able to detect each one in each individual. And so trying to monitor that population over time is a good way to go. And really, this community wastewater represents a snapshot, if you will, of the status of public health in a certain community. So on this slide, you can see a variety of the different viruses in particular that have been analyzed through wastewater. And the primary method for all of these is laboratory detection using polymerase chain reaction. And of course, the coronaviruses are there, including SARS-CoV-2 as well. So this was done for other coronaviruses. And you can see a variety of different types of viruses here, both RNA as well as DNA viruses are on the slide. And if we look at some of the other studies that have been done actually using metagenomic analysis to detect various viral sequences that were found in wastewater, there's a large variety of viruses that have been looked at in terms of their sequences that have been found. And it's, again, a really interesting approach to looking at community analysis. And you can see in just some of these samples, the wide range of viruses that could be detected. So, of course, this approach to wastewater-based epidemiology has motivated many public health scientists around the world. And a lot of this has been methods driven, right? Trying to understand what is the best way to come together as a community to make sure that we're doing the best analysis that we can, and then be able to try to compare that from country to country and location to location. And here you can see some of that information represented on this figure here when thinking about some of the differences between urban and rural communities. Where do you collect that data? What other data is important alongside those initial samples? Are we thinking about how that population is impacting it? Are we using certain biomarkers? Are you going to figure out from census tract how many people are there? And, you know, many times if we think about rural communities, there are many people who are not on a municipal water system. And so it's more difficult to be able to look at those communities. So what are we doing if we're leaving out those communities? I think that's another critical piece of understanding. And then thinking about what models we apply to all of these so that we can understand what's happening in these outbreaks. So there is a lot of work looking at population normalization, and that's very important when it comes to thinking about disruption that might happen within climates, and when thinking about weather, also thinking about populations. As I mentioned before, many scientists are looking to census tract information, but there's some literature suggesting that that's not necessarily the best way to go. But if we use adjustments for human population through different biomarkers, that's better than looking at government census tract data. And here you can see some of the different biomarkers that could be used in this analysis. So how do we use the information now to support the current COVID-19 pandemic? And so I'm going to show you some specific data that comes from my lab as well as around the world. And as we've all looked to try to understand the spread of SARS-CoV-2. Here's a picture of the University of Delaware campus and some of my group outside of one of the residence halls trying to place one of our samplers one day and making sure that we were getting the best sample that we could. And really, this information is very useful, but it's more useful when it's used as a complementary approach to clinical testing. And so I think that piece is really important, especially in small catchments. As I mentioned, this has been going on around the world. Some of the groups, such as these Dutch scientists here in the middle, and others around the world, were some of the first to be doing this early on in 2020. And it's been very exciting to see how much people are communicating, how much sharing is going on in terms of making sure we're all using the best approach that we can. And of course, we're using PCR when it comes to detection from the wastewater. And so this is a great paper that looked at what was being done early on in the pandemic, and making sure that many of the early adopters of this technology and early publications were using complementary approaches. And so that the rest of us learning from them would be able to do the same. This is the, I guess this was taken probably March 1st or so. So a few days old. There's probably a few additional groups that were added onto this, but this is the COVID-19 WBE Collaborative, which is an incredible organization. They're on social media platforms and online, and there's the website here. You can visit them and you can see all the different groups that are involved in this. And these blue markers just continue to grow by the day. There's more and more of them all the time. And again, thinking about the methods that are involved in this, we have to think about what communities we're sampling and what's the size of these communities. In most cases, people are looking at larger municipalities, but now we're even looking at smaller catchments, thinking about hospitals and schools with the drive to reopen schools in many places around the world. How can we use this to help inform those decisions? And here you can see that there's this potential for wastewater monitoring to inform the surveillance, the epidemiological models, and as well as risk assessment. And if it was only this easy, right? If only we were able to run the water through a simple filter, exclude all the larger particles and collect out the viruses. Well, we know that's not as easy as it is. And when we think about what else is in wastewater, we know we're going to have inhibitors that are there that may result in the loss of some of those viruses. And so pretty typically, while there are still different ways to do this, we're looking at a filtration step, a concentration step, an extraction step, and a detection step. And in most cases with the wastewater, there's a pasteurization step to start off. So a heat treatment at 60 degrees C for about 60 minutes, just to reduce the risk of working with these samples. And this is an example of one of the workflows that has been designed. Again, the response from industry has been very quick in terms of development of different tools and diagnostics. And here you can see an example of QIAGEN and the platform that they have where you can work straight through. And they even have an addition onto this that you can use for detection of the variants of concern to be able to look to sequence what you find in wastewater. And so this is the platform that we are using, and we've been very lucky to work with some excellent people at QIAGEN. From the same paper that I showed earlier, they pointed out some really unique questions that go along with this wastewater sampling. For example, different from looking at a river, right? When we think about flow, if I look at a river, we have a constant flow of water. But if I look at these sewer sheds, I don't always have a constant flow of water, depending on how many people might be in these residence halls, for example, shown in this top right picture. And we think about people's habits. Bathroom habits are an intriguing thing when you think about schools or hospitals. And so what's the best time to collect? So most of the time we are taking a 24-hour composite sample, but there are some cases when a grab sample would be just as good and perhaps more efficient in terms of people's time. And so thinking about the type of sample that's collected is really important for that given community. So here on this graph, I'm showing you three different catchments. One is a very large wastewater treatment facility, and that is within the city of Wilmington. This represents about 500,000 people, and here you can see differences in terms of the virus that was collected. And this is all in viral copies per liter over time and goes into early February. And of course, as we've seen around the world, when we had some increases around holiday time, and we had some, decreases where people were really following those mitigation efforts and good behavior. And then if we go to a smaller catchment that only has about 400 people, we see lower virus presence there, but then it sort of mimics when we think about what's happening with those trends. This is at Christiana Hospital, which is a large hospital center in Newcastle County in Delaware. And here you can see some variability, and we actually are working with some analysts who are trying to understand how to utilize this data, in particular when people were very concerned about what flu season might be like, to understand what their beds might be like. Do they need more beds? What is the virus like in the community? Are we going to have more and more people coming in with COVID-19? Or are we going to have room for people who might have flu? So good questions that can be answered with a lot of this analysis. Here is an example of a chart that we put together looking at the different levels of viruses that were detected in 4 different wastewater treatment facilities. And again, there's some difference if we're looking at these treatment plants or if we're looking at pump stations. The analysis can be a little bit different. And then we can put this all together and see within these larger wastewater treatment plants, the change that occurred over time. And it's sort of reflective for all of us to see where we've been through, right? And I think we're all going to be wrestling with lots and lots of data for the next several years, unfortunately. And you can see here, we're getting into the very high amounts as we get into December. And then if we were to keep playing into February, we would see those starting to come down. And so that's the good news of those trends. This is what the dashboard looks like, where we house this data. And people are using this in real time. And again, there are multiple laboratories that are doing data analysis to try to make sense of what's happening across this county. And in particular, as I mentioned, working with hospitals as well as nursing care facilities and other places, schools, those kinds of things. So it's good to see that this is being used alongside the rolling day clinical averages here, like that's what's shown here on the top right-hand side. So again, this is a really important tool in our toolbox. It's a complementary approach to surveillance. We think about people who are asymptomatic and pre-symptomatic and may not be identified through testing. This is a good way to identify them. We can look for evidence and reemergence of some of the different strains as people are doing more and more sequencing on wastewater analysis. It also provides useful information if we need to pivot in terms of our approach for mitigation. Lots of questions that still remain, including we don't understand the variable shedding dynamics of different individuals. We need to know more about what dilution is like and how the effect of weather events affects our wastewater, and also the use of different indicators and biomarkers is very important. And of course, none of us have this crystal ball, so we don't quite know what's going to happen in the future, but hopefully we all stay on a good trend. And with that, I want to thank you very much for this opportunity and remind you that on our IAFP website, we have some great resources for you, and please stay vigilant, stay optimistic, and thanks again.